Good morning, everyone. Today, I want to take you on a journey into one of the most fascinating and elegant structures in the human brain, the cerebellum. While it constitutes only 10% of the brain's volume, it contains more than 50% of all neurons in the central nervous system. Far from being a simple coordinator, the cerebellum is the brain's master driving instructor for all movements. It fine tunes our balance, posture, eye movements, and every skilled coordinated action we perform, from walking smoothly to playing a musical instrument or performing a precise neurological examination. In this talk, we will explore number one, the core functions of the cerebellum, number two, how it acts as an accelerator, brake, and clutch in the motor control, number three, its beautiful internal circuitry involving the Purkinje cells, deep cerebellar nuclei, climbing and Mossy fibers, number four, the critical role of the motor error correction and motor learning through the long term depression, which is abbreviated as LTD. Number five, its anatomical divisions and connections. Number six, finally, how we clinically test the cerebellar function. So let's begin by understanding what cerebellum really does in our daily life. This is the title slide, the cerebellum, the brain's master driving instructor, accelerator, brake, clutch, and motor learning. The cerebellum is basically divided by different methods. One method is the anterior lobe, which is separated from the larger posterior lobe by the primary fissure, which you see here. The posterior lobe is divided from the smaller flocculonodular lobe by the posterolateral fissure. Another method is its division into the vermis, paravermis, and cerebellar hemispheres. The cerebellar vermis, it receives mainly the proprioceptive input from the trunk, neck, and head, and the paravermis, it receives the from the extremities, especially the proximal extremities of upper and lower limbs, while the cerebellar hemispheres receives information from everywhere up in the brain, down in the spinal cord, and as well as the peripheral sensory receptors. Anterior lobe, which is vermis and paravermis, is also known as cerebellum. It is mainly the receptive center from the proprioceptive pathways. Posterior lobe, also known as the cerebrocerebellum or cerebellar hemispheres, it receives projections from the primary and supplementary motor cortex, premotor area, primary sensory cortex. The flocculonodular lobe, also known as the vestibulocerebellum, has to and fro connections from the internal ear, modulating the vestibulo-ocular, vestibulospinal, and vestibulo-colic reflexes. There are four main nuclei which are in the white matter of the cerebellum. From lateral to medial, they are dentate nucleus within the cerebellar hemispheres, amboliform and globose nucleus, which are respectively called anterior and posterior nucleus, and combined they are called the interpositus nucleus that lie within the vermis and paravermis. And last is the vestigial nucleus that lies within the flocculonodular lobe and vermis. So, from lateral to medial, these can be remembered by the acronym "Don't eat greasy food." That's how I remember. The cerebellum is a master driving instructor for motor skills. What does it functions? Balance, equilibrium, posture and eye movement, coordination of half automatic movements of walking and posture maintenance, muscle tone adjustment, motor learning and motor skills. This is a very important function. It receives fibers to and fro from the inner ear, proprioceptive informations from all throughout the body. It has connections with the primary sensory and motor cortex, to and fro loop, and this information is also received from the muscle spindles, is also sent to the cerebral cortex for modulation. So think of cerebellum as an automatic skill sharpener that helps you perform the smooth, precise, and well-timed movement, just like learning to drive a car perfectly. The deep cerebellar nuclei and various cells act as accelerator, brake, and clutch in this process. So this is very important. The cerebellar cortex consists of three important layers, which from outside to inside are the molecular layer, Purkinje layer, and granular layer. The molecular layers basically contain the axons of the Purkinje cells. The Purkinje layer contains the cell body of the Purkinje cells, while the granular layer contains the Golgi cells and the granule cells. Most of the input to the cerebellum, output to the cerebellum, goes out through the deep cerebellar nuclei, but this information is heavily and totally modulated by the Purkinje cells. The Purkinje cells, it receives information from the contralateral olive via the climbing fibers that secrete aspartate, which is an excitatory neurotransmitter. When it releases aspartate at the Purkinje cell, the Purkinje cell, it gets excited and releases the GABA. So GABA is an inhibitory neurotransmitter, and this is resulting in the error connection or neural sharpening when the climbing fibers and the parallel fibers that we'll see right now, they together excite the Purkinje cell. The sensory pathways are through the Mossy fibers, which send their axons to the deep cerebellar nuclei, as well as the granule and cells and Golgi cells in the granular layer. And from the granule cells, the axons, they relay to the molecular layer, sending the parallel fibers, which are important. They relay information all throughout the cerebellar cortex. These parallel fibers, they also send axons to the Golgi cells, which first excites the Golgi cells, and the Golgi cells then inhibit the granule cells. Likewise, in the molecular layer, the axons are sent by the parallel fibers to the stellate cells and the basket cells. The stimulation of these cells, that is the basket cells and stellate cells, is inhibitory to the axons of the Purkinje cells, and inhibition of the Purkinje cells in fact means disinhibition, that is that the cessation of the secretion of the GABA. But however, it also sends the excitatory neurons to the Purkinje cells. So there is a two-way system. Timing of motor activity and progression of one muscle group movement to the next are the main functions of the cerebellum. To sum up, how does cerebellum does all this? It receives information from the cerebral cortex and from the peripheral sensory receptors like muscle tendon organs, Golgi tendon organs, etc. It compares the two, that is the intended movement and actual movement. Correction of errors in the motor activity, it results in motor learning. Plans the next sequential movements, learns from the previous errors made by it. This is a very important circuit, inferior oblique in motor error connection. This is a hand, the index finger of which want to touch this object. So this is the intended movement. As far as the intended movement occurs without error, there is no activity in the olives. As soon as the intended movement makes a motor error, as shown here, the olives are activated, and it results in sending signals via the climbing fibers and the parallel fibers that trigger the complex and the simple spike respectively. The complex spike is a powerful error signal or teaching signal carried out by the climbing fiber system. It plays a central role in both immediate motor correction and inducing long-term depression at the parallel fiber Purkinje cell synapses for motor learning. So when the climbing fibers and parallel fibers, they concurrently act on the Purkinje cell, it leads to long-term depression. And finally, a corrected motor output is sent to the deep cerebellar nuclei. The long-term depression or LTD at the parallel fiber Purkinje cell synapses, how it is defined, it is a persistent decrease in the synaptic strength. It occurs at the parallel fiber Purkinje cell synapses. Parallel fibers are the granule cell axons. Purkinje cells are the principal cerebellar neurons. Long-term depression was first described in early 80s by Masao Ito and colleagues. It is based on paired stimulation of parallel fiber and climbing fibers. It results in long-lasting depression of the parallel fiber responses. Low frequency 1 to 4 Hz paired parallel fiber climbing fiber stimulation. Parallel fiber alone or climbing fiber alone usually insufficient to cause long-term depression. Climbing fiber provides a teaching or error signal. There is a heterosynaptic pathway. The climbing fiber input depresses the parallel fiber synapses. Occurs on same Purkinje cell. Lasts for hours or longer. That results in long-term plasticity. Long-term depression is bidirectional with long-term potentiation. Long-term potentiation can reverse long-term depression under different conditions. Deep cerebellar nuclei are the accelerator or the main driving force. These are the final output stations of the cerebellum. They send excitatory signals to the ventrally located motor thalamic nuclei and brainstem to drive the movement. Without them, movements would be clumsy. What is their role in sharpening? They provide the raw power and drive for intended movement. The cerebellum constantly tunes how hard or how softly this accelerator is to be pressed. Purkinje cells, the brake or the primary inhibitory control. These are the only output neurons of the cerebellar cortex. In fact, why I am telling it only output neurons, because without the influence of the Purkinje cells, the deep cerebellar nuclei cannot send its projections outside the cerebellum. All the projections from the deep cerebellar nuclei needs to be modulated by the Purkinje cells before they are being sent. They release the inhibitory gamma aminobutyric acid and powerfully inhibit the deep cerebellar nuclei. They act as the main brake by inhibiting the accelerator, that is the deep nuclei. They sculpt and refine the movements, preventing them from being too jerky, resulting in overshoot or undershoot or uncoordinated movement. What is their role in sharpening? Strong Purkinje braking implies smooth, precise, and error-free movement. Weak or mistimed braking results in ataxia, tremor, and dysmetria. Climbing fibers from the inferior oblique, which act as a clutch plus error teacher. These are the powerful one-to-one excitatory inputs using aspartate that powerfully activates the Purkinje cells. They carry error signals implying that something went wrong in the movement, like pressing the clutch to disengage gear and allow adjustment. Climbing fibers temporarily interrupt the ongoing pattern and trigger plasticity by long-term depression at parallel fiber Purkinje synapses. What is their role in motor learning? They teach the cerebellum that this movement needs correction. Repeated error signals help sharpen the skill over time. Mossy fibers, these are the main information highways in terms of context and sensory input. In the epic story of Hindus, Mahabharata, Sanjay was granted the divine vision to perceive events of battlefield in real time and relay them directly and accurately to Dhritarashtra, who was blind since birth. So, Mossy fibers carry diverse high volume real time inputs from the multiple sources: spinal cord, vestibular system, and the cerebral cortex. They provide the cerebellum with a comprehensive state of the body and plant movement, much like Sanjay provided a complete situational awareness of the battlefield. They excite granule cells and also send collaterals directly to the deep nuclei. They provide the information what is happening now data that system uses to decide how much accelerator or brake is needed. Just as Dhritarashtra depended entirely on Sanjay's input, the cerebellum depends heavily on mossy fibers for accurate context before fine tuning the movement. Unfortunately, the blind king was unable to control his generations. However, the cerebellum does it so nicely with the help of the information it receives through the mossy fibers. Granule cells, they are the distributors or gear multipliers. They are the most numerous neurons in the brain. Underline this. They receive mossy fiber input and send parallel fibers that excite thousands of Purkinje cells. They spread and amplify the contextual information widely. What is their role? They turn one input into a broad signal that allows fine spatial and temporal sharpening. So that is like the making a WhatsApp message viral on several WhatsApp groups. So this allows a broad signal into whole cerebellum. Golgi cells, the gain controller or the feedback brain on the granule cells. They inhibit the granule cells by negative feedback, prevent overexcitation, and control the strength and duration of the mossy fiber signals. What is their role? Like adjusting the sensitivity of the accelerator pedal so it doesn't flood the system. Basket and stellate cells, the lateral sharpeners or the focal brakes. They are the inhibitory interneurons in the molecular layer. They provide the lateral inhibition to the Purkinje cells. They sharpen the signal by inhibiting the neighboring Purkinje cells, creating a contrast like edge detection. What is their role in sharpening? They make the braking pattern more precise and focused, preventing diffuse or sloppy movements. Cerebellar afferents, they are from the cerebral cortex, cerebropontocerebellar tracts, cerebro-olivocerebellar tracts, cerebro-reticulocerebellar tracts, and from the peripheral receptors, anterior and posterior spinocerebellar tracts, cuneo-cerebellar tract, and vestibulocerebellar tracts. Now we come to very important cerebellar connection slide. This is the cerebellum, the hemispheres. It is connected to the brainstem by the superior, middle, and inferior cerebellar peduncles, which has afferent and efferent connections to the other parts of the brain spinal cord. The middle cerebellum has got only the fibers which come from the cortex to the cerebellar hemispheres. So we start with the superior cerebellar peduncle. Here, efferent pathways are mainly the dentatothalamic and dentatorubothalamic and cerebellovestibular tract. The dentatorubothalamic and dentatothalamic tracts. The dentatothalamic tract originates from the neurons in the dentate nucleus, the largest of the deep cerebellar nuclei, located laterally in the cerebellar white matter. It leaves the cerebellum via the superior cerebellar peduncle, ascends through the upper pons and midbrain, decussing in the midbrain, and terminating primarily in the contralateral ventrolateral nucleus of the thalamus. It relays cerebellar output to the motor and premotor cortex via the thalamus for planning, coordination, and execution of the voluntary movements. dentatorubro-thalamic tract, which starts from the thalamus here, goes to the red nucleus and then relays information to the thalamus. It is originating from the dentate nucleus of the cerebellum. It follows the ipsilateral superior cerebellar peduncle, deposits in the midbrain near the inferior colliculus levels. It reaches the contralateral red nucleus and then continues to the contralateral ventrolateral thalamus before relaying to the motor cortex. The functions of the dentatorubro-thalamic tract: it functions to transmit the cerebellar signals to the red nucleus and thalamus. It modulates the motor control, muscle tone, and skilled voluntary movements. Cerebellovestibular tract: it traverses via the superior cerebellar peduncle as well as from the inferior cerebellar peduncle. This portion of the cerebellovestibular tract originates from the fastigial nucleus, particularly its caudal and rostral part that send axons involved in the vestibular and postural control. Some of these fibers initially travel or loop over the superior cerebellar peduncle before crossing or joining the uncinate fasciculus, that is the hook bundle of Russell, then descend to reach the contralateral vestibular nuclei and reticulo formation via the roots that ultimately utilize the peduncle, inferior peduncle components for final termination, influencing the bilateral vestibular outflow. It modulates the vestibular nuclei and reticular formation to regulate the posture, balance, and bilateral vestibular reflexes. So from here, these tracts they go further into the spinal cord as the vestibulospinal tracts. Tactocerebellar tract. So we come to the efferent of the superior cerebellar peduncle. The tactocerebellar tract originates from superior and inferior colliculi in the tectum of the midbrain, carrying the visual and auditory information. It enters the cerebellum via the superior cerebellar peduncle ipsilaterally and terminates mainly in the vermis and paravermal regions of the anterior and posterior lobes. It conveys the visual and auditory information from the midbrain tectum to the cerebellum for integration in orienting movements and multisensory coordination. The rostral cerebellar tract, which is not shown here, and the ventral spinocerebellar tract, they are important. The rostral spinocerebellar tract originates from the neurons in the intermediate gray matter, that is the laminae 5 to 7, and the ventral horn of the upper cervical and thoracic spinal cord, primarily C4 to C8 levels serving the upper limbs. It ascends ipsilaterally in the lateral funiculus, enters the cerebellum via the ipsilateral inferior or superior cerebellar peduncle, and terminates in the ipsilateral anterior lobe vermis or paravermis as upper limb equivalent of the ventral spinocerebellar tract. It provides the unconscious proprioceptive feedback from the upper limbs to the cerebellum for fine coordination and correction of the ongoing movement. The ventral spinocerebellar tract, it arises from the muscle spindles. In fact, the muscle spindles sends the projections to the spinal border cells in the laminae 5 to 7 of the lumbosacral spinal cord, carrying the proprioceptive information from the lower limbs. Its fibers cross the spinal cord and then ascend contralaterally in the ventrolateral funiculus through the brainstem and enter the cerebellum via the superior cerebellar peduncle and mostly recross to terminate ipsilaterally in the vermis and the paravermis of the anterior lobe. So here from the muscle spindle it goes to the lamina 5 to 7, crosses, and in the lateral funiculus it reaches high up and it recrosses again and enters the superior cerebellar peduncle and ends on the granule cells here. What does it function? It carries the proprioceptive and motor efference information from the lower limbs to the cerebellum for real-time monitoring and correction of the locomotion and correction. Now we come to the corticospontocerebellar tract, which receives information from the cerebral cortex, shown in the blue here. The fibers they go to the pontine cerebellar nuclei and cross to the opposite side and enter the cerebellum to via the middle cerebellar peduncle. There is also another pathway. The From the cortex, the fibers they reach the ipsilateral inferior oblique, and from here they climb as climbing fibers and reach the Purkinje fibers. So basically, it is cortico olivo cerebellar tract as well as the cortico pontocerebellar tract. The cortico-pontocerebellar tract originates from the widespread areas of the cerebral cortex, primarily the frontal motor, premotor, parietal, and other association areas. Corticopontine fibers descend ipsilaterally through the internal capsule and cerebral peduncle to synapse in the pontine nuclei. Pontocerebellar fibers then decussate in the pons and cross via the mid-cerebellar peduncle to reach the contralateral cerebellar cortex, mainly the posterior lobe or neocerebellum, and dentate nucleus. It transmits the planning and command signals from the cerebral cortex to the cerebellum, enabling the cerebellum to compare the intended versus the actual movements for smooth motor performance. The dorsal spinocerebellar tract here originates from the Clark's column, that is the nucleus dorsalis in the thoracic and upper spinal cord from T1 to L2, receives the proprioceptive input from the lower limbs and trunk via the muscle spindles and Golgi tendon organs. It ascends ipsilaterally in the dorsolateral funiculus, enters the cerebellum via the inferior cerebellar peduncle, and terminates ipsilaterally in the vermis and paravermis of the anterior lobe. How it functions? It delivers the unconscious proprioceptive information from the lower limbs and trunk to the cerebellum and coordinates the posture and gait. The cuneo-cerebellar tract, which is not shown here, originates from the accessory, that is the lateral cuneate nucleus in the medulla, which is proprioceptive input from the upper limbs and trunk via the cuneate fasciculus, ascending ipsilaterally as external arcuate fibers and enters the cerebellum via the inferior cerebellar peduncle to terminate ipsilaterally in the anterior lobe for the upper limb representation as the upper limb equivalent of the dorsal spinocerebellar tract. How does it function? It conveys the unconscious proprioceptive feedback from the upper limbs and neck to the cerebellum for precise coordination of the arm and hand movements. Now comes the vestibulocerebellar tract. The vestibulocerebellar tract, which is afferent, originates primarily from the vestibular ganglion, the primary fibers via the 8th cranial lobe, and secondarily from the vestibular nuclei in the medulla and pons. It travels via the juxta restiform body, which is a part of inferior cerebral peduncle, and projects mainly to the ipsilateral flocculonodular lobe, uvula and uvula, to carry the vestibular information for balance and eye movement. What are its functions? It provides the primary vestibular input for the cerebellum for maintenance of balance, equilibrium, and coordination of eye movements for head position. Olivocerebellar tract is very important, which I have already shown here previously with the cortico-pontocerebellar fibers and the cortico-olivocerebellar fibers. The olivocerebellar tract originates from the inferior olivary nucleus in the medulla. Its climbing fibers, they decussate in the medulla here, enter the cerebellum via the contralateral inferior cerebellar peduncle, and terminates as climbing fibers on the Purkinje cells throughout the contralateral cerebellar cortex. How does it function? It delivers the error signals and timing information via the climbing fibers to the Purkinje cells, essential for motor learning, adaptation, and correction of the motor errors. In fact, this fibers, all these fibers which are going as climbing fibers are also receiving input from the muscle spindles here. It goes to the olive, and then it crosses opposite side and reach as climbing fibers via the inferior cerebral peduncle to the Purkinje cells. Reticulocerebellar tracts, they arise from the reticular formation, which is shown here in the blue. It originates from the neurons in the pontine and medullary reticular formation, including the paramedian reticular nuclei. It enters the cerebellum mainly via the inferior cerebellar peduncle and projects bilaterally, predominantly ipsilaterally to the vermis and the vestigial region as Mossy fibers carrying multisensory and motor planning information. So it is so important taking information from every part of the brain. It relays the multisensory state dependent information from the reticular formation to the cerebellum for integration in posture, locomotion, arousal related motor control. Cerebellovestibular tract. These are the efferent tracts. Some of them they are going through the cerebellovestibular tract originates from the vestigial nucleus and to a lesser extent the interposed nucleus of the cerebellum. It exits via the inferior cerebellar peduncle, that is the uncinate fasciculus or the juxta-restiform body, and projects to the contralateral and some ipsilateral reticular formation in the pons and medulla to influence the reticulospinal tract and posture. What is its function? It sends cerebellar output to the reticular formation to modulate the reticulospinal tracts, thereby influencing the axial and proximal muscle tone and posture. Now, finally, the cerebellovestibular tract. The cerebellovestibular tract shown in the green here originates mainly from the flocculonodular lobe and vestigial nucleus of the cerebellum. It exits primarily via the inferior cerebellar peduncle, juxta-restiform body, and projects to the ipsilateral vestibular nuclei. Here it is shown here in the medulla and pons to modulate the vestibulospinal and vestibulo-ocular reflexes. How vestibulo-ocular reflex? Because it's now communicates here with the medial longitudinal fasciculus that is connected to the third, fourth, and sixth cranial nerve nuclei. What does it function? It provides the cerebellar feedback to the vestibular nuclei to the fine-tune the balance, posture, and the vestibulo-ocular reflex for stable gaze during the head movements. So finally coming to the clinical testing of the cerebellar function, which is shown by a video here where I am testing a normal individual for the cerebellar functions. I am going to describe you the examination of the cerebellar system. The cerebellar system is examined in the eyes, in the limbs, upper limbs and lower limbs, as well as in the trunk. Historically, a patient with cerebellar ataxia has got a wide-based stance. Means if he is asked to put his feet together as a group, he sways. To make the base more stable, he has a wide-based stance like this. So this is one of the things. The second thing is that in the eyes there are many abnormalities. First is the examination of the smooth pursuits which become the saccadic pursuits. So the pursuits, as I told in my earlier video, that are to be examined. The distance which is to be tracked by the patient's eyes has to be at least 50 centimeters away, which is here, and I am tracking his eyes from side to side horizontally, and here I will see the gaze holding while moving the eyes around 20 to 30 degrees. The full range of around 40 degrees would evoke or can evoke endpoint nystagmus, which is difficult to differentiate it from the gaze evoked nystagmus. The speed of the target which the patient is following should be very slow. So this is in the horizontal plane. Likewise, in the vertical plane, it is tested up and down. So this is around the smooth pursuits. For the seconds, I ask him, इधर देखो, टू शिफ्ट हिज गेज, कैमरे की तरफ देखो, मेरी नाक की तरफ देखो, कैमरे की तरफ देखो, कैमरे की तरफ सामने देखो, मेरी नाक की तरफ देखो, कैमरे की तरफ देखो. So saccadic hypometria or hypermetria may be present. The hypometria occurs in the dorsal vermis lesions, while the ventral vermis lesions causes the hypermetria. The third is VR suppression. उंगली ऐसे रखो, इस उंगली की तरफ देखते रहो, जहां गर्दन जाएगी वहां देखते रहना. We see is he able to suppress the vestibulo-ocular reflex normally or not? He is able to do that. So the three main examination things for cerebellum is examined. The abnormalities could be gaze-evoked nystagmus, which occurs because of the lesions in the flocculus and paraflocculus, or the saccadic pursuits movements, or there can be a hypometric or hypermetric saccades. The second examination consists of the limb ataxia. ये उंगली ऐसे करो, एक बार मेरी उंगली पे लगाना, एक बार नाक पे लगाना, इसको इस लेवल पे पकड़ लो. अब लगाओ, नाक पे लगाओ, तेरी उंगली जल्दी जल्दी लगाओ, तेजी से, इसको रोटेट करके लगाओ, यू और यू, तेजी से गर्दन. The target should be moving and should be made much more difficult. अब दूसरे हाथ से करो. We do it from each side. इसको इस लेवल पे रखो. अब जल्दी जल्दी. So this is the limb ataxia. The third is dysdiadokinesia. ये हाथ ऐसे करके तेजी से ऐसे ऐसे करो, और तेज करो, और तेज करो, और तेज करो. अब दूसरे हाथ से ऐसे ही करो. For the lower limb in coordination, we are testing the appendicular ataxia. लेट जाओ उधर वहां पे. हाँ, ध्यान से देखना. ये एडी अपने इधर घुटने के नीचे ले जाके इसको रगड़ना है, यहाँ तक तेजी से यूं उधर सर्कल लेके आना. ठीक है, ऊपर ले जाओ, ऊपर यूं. दिस इज द heel shin test. Likewise we test for the other side, heel shin test. तेजी से करो. The other test for testing in coordination in lower limbs is the toe finger test. ये मेरी उंगली पे लगाओ, तेजी से नीचे करो, लगाओ. You can make the target complex. मैं लगाओ, लगा लो. अब उस पैर से करो. So this is about the in coordination in lower limbs. Now comes the trunk. For trunk there are two things. बैठो, इधर निपटा, नीचे कर दो. One, you need to observe whether there is a truncal neck titubation like this when the patient walks, and secondly, the truncal ataxia can be assessed by making the patient to balance only on his trunk. ये पैर ऐसे यूं करो, ऐसे हाथ से पकड़ो, दूसरा वाला भी ऐसे करो, उस हाथ से पकड़ो, इसको ऊपर रखो, उधर साइड से टेक फ्रॉम दिस साइड, यूं थोड़ा आगे सरको कि थोड़ा हवा में हो, हवा में. Yes. So now he is simply on his trunk, balancing his trunk on his bottoms. So this person's truncal coordination is perfect. So neck titubation and ability to balance your bottoms, that is your trunk on your bottoms, without the help of upper or lower limbs is the test of coordination of the trunk. That's all. Thank you very much. One of the important tests to assess the midline cerebellar ataxia is the tandem Romberg's test and tandem walking. The tandem Romberg's is carried out by asking the patient to stand tandem for around 30 seconds. If the patient is able to do that, then he is asked to walk tandem, say around 10 to 12 steps, and if possible, backward 10 to 12 steps. अब इसी तरह से एक कदम आगे रख के चलते रहो, चिपका के। पास रुक जाओ, अब पीछे चलो ऐसे चिपका के। So this completes our cerebellar testing. Thank you very much. So now I finish my talk here on the cerebellum. Thank you very much.